Okay, it's April the 12th. This is the genetics class. We have one student filming and one person who is absent because we don't want to have 15. Okay, the lesson that we're going to be doing today is showing us a comparison between a very common process that happens in biology when your DNA makes a copy of itself. What's that called, Vanessa? Replication, that's right. So, replication, sometimes there's a lot of molecular biology happening and it's hard to visualize what all takes place. So this group is going to model replication. They have instructions on what they need to do and what processes they need to demonstrate. Then we're going to move to a higher tech table. This is going to be the PCR, which stands for what, Ramona?
Polymerase chain reaction.
Very good, Ramona. Polymerase chain reaction. And this table is going to demonstrate how by using a piece of technology called what, Sue?
Thermocycler.
A thermocycler, that's right. Using a thermocycler, we're going to be able to take pieces of DNA and amplify them. So this table will be doing PCR. This group will be working on programming the thermocycler because some of the labs we are doing, thanks to the Princeton Loaner Kit, we are able to actually amplify our own DNA. We have done the ALU insertion lab, and we're going to be doing the PTC lab, checking to see if we have the allele for the bitter tasting gene. So this group will be working on learning how to program and also temperature conversions, and if you look at the bottom of your sheet, determining how many copies of your amplicon will be produced after a certain number of cycles. Okay, so does everybody have their sheet and understand their directions? And then as you're working on it, you should also have one sheet from your group where you're going to be comparing replication, PCR, and both, and then proteins, nucleic acids. Sometimes those things, it's almost like we memorize what they're used for, but we don't visualize it. The purpose of this lab is to help you to visualize and hopefully to clear up some misconceptions. So if you'll go ahead and get out your pieces of DNA, and when Nesimo gets this open, remember you're only supposed to have one piece put together. The other should be assembled in nucleotides. Ethel, y'all can go ahead and start breaking that into nucleotides. And Leon, what three parts are there to a nucleotide?
Phosphate, sugar, and a base.
Okay, let Leon hold that and point those out. Okay, hold it up. I don't know if the camera can see it. Okay, point out the three parts for us.
That one, that one, that one.
Okay, does he have anything extra on there that he doesn't need?
Extra. Base.
What do y'all think, Fruity?
What do you think, Abby?
Where's your nitrogen base? Okay, the blue. Where's your sugar?
The black.
The black.
Okay, and then where's the phosphate?
The white.
How many does he have on there?
He has two. He has three.
Okay, before it attaches, it does have three, but these are already part of the model of DNA, so they only have one. So, what does he need to do?
He needs to take off one.
Okay.
If I can get it off.
That's why we got a strong man like you, Leon. Now,
Yeah, we have.
looking at this, Ethel, let's see, do y'all have the phosphate on the correct carbon? See, sometimes in molecular biology you start hearing about this 3', 5' carbon. How many carbons are in the sugar, Fruity?
5.
Okay, which carbon is the nitrogen bond attached to?
3.
Okay, is it number 3, the nitrogen? It's number 1.
I meant 1.
Okay, so is the nitrogen attached to number 1? Now, is the 3' or the 5' supposed to be the free end? Which one's the free one? Ready for attachment. Do y'all know?
I think it's a three.
Three.
Think it's the 3? Is that what you're going to go with, Petunia? The 3? Okay, is your 3' end, is it free and ready for attachment?
Yes.
Okay, let's look. Isn't this going to be the side of DNA? So this is in the middle, and this is number 1, 2. What's that one?
3.
3. So is it free, or do we have our phosphate on
We
it?
have phosphate on it.
Okay? So, the 3' end is the one that's open, ready for our new nucleotide to attach to it. And see, it's hard to see that sometimes even with a model. You got to orient it. Let me see if this group back here has their free nucleotides arranged correctly. Okay, let's give Matilda one. Just a nucleotide. Okay, hold it up for us, Matilda. Okay, now, hold it like it's being oriented in the DNA ladder, okay? Is her phosphate on the correct carbon? Where does it need to be? Well, if she flips it like that, that's right. So it depends on how you have it oriented, doesn't it? That's a quick way to solve the problem. Okay, good. Y'all go ahead and you're working on your PCR. Okay, over here at the thermal cycler, okay, what are we doing?
We're working on our temperature conversions.
Okay, and how many different temperatures do you have to go through when you do PCR?
You have to go through 3 temperatures.
3 temperatures, okay? The first temperature is what, Sue?
94 degrees Celsius.
94 Celsius, okay? And did we remember how to convert from Fahrenheit to Celsius?
We didn't.
Okay. Well, let's think about it. What kind of ideas did y'all have for doing your conversions?
Mavis, didn't you think of something?
Okay, what'd you think of, Mavis?
What'd you think of it?
See?
Janika.
Oh, wow. I know there's a 32 somewhere. Maybe you added or subtracted, and a 1.8, I think.
Okay. It's amazing how shy y'all are for the camera. Okay, let's see. So you know something about a 32, and you know something about 1.8, and I sort of remember that too. Okay. Well, let's just think back for a minute. Celsius to Fahrenheit, or Fahrenheit to Celsius. We're going Celsius to Fahrenheit. Should the number get bigger or smaller? Okay. Eunice has a good idea. She's setting up a temperature scale and she's identifying our 2 markers, temperatures. When does water freeze? Celsius.
At 0 degrees Celsius.
And Fahrenheit, it's what?
32.
So when I just asked that question, if you're going from Celsius to Fahrenheit, should it get larger or smaller? Go back to 2 reference points that you know, and you can see that if you're going to Fahrenheit, what should the number do?
It should increase by 32.
Okay, so it should get larger. What about from when water boils?
It's going to increase 112.
Okay, so it definitely increases. So we know it's going to increase. Okay, so Celsius to Fahrenheit. We know then, are we probably going to multiply or divide by 1.8? Because we got to get bigger. And then are we going to add or subtract the 32? We're going to add it. Okay, so you're on the right track. Now see if y'all can find the rest. I'm going to check on the other group. I'll be back in just a minute. Okay, tell us what's going on over here, Vanessa.
Okay. Right now, helicases just came and separated the double-strand DNA.
Okay, let me ask Leon with his cute little pink bracelet. What does that represent, Leon?
Helicase.
Helicase. Okay, how come it doesn't look like this and have these little structures?
It's not that.
Okay, what is it?
It's an enzyme.
It's an enzyme. Okay, which organic class does an enzyme belong to? Proteins, that's right. Now, which organic class does DNA belong to?
Nucleic acid.
Nucleic acids. So usually we learn about 4 different classes: lipids, proteins, carbohydrates, and nucleic acids. But sometimes it helps if you can sort of see their basic structure. So what's the basic structure of DNA? What is one little monomer called?
Nucleotide.
Nucleotide. Now what is one little monomer called on a protein?
Amino acid.
Okay, good. Okay, so what'd you say helicase does, Leon?
It opens up the DNA
Okay, and what's the purpose? Why even bother opening it
so
up?
you can copy it.
So you can copy it. So we can't copy it if it's closed? No. Okay, no copying if it's closed. Okay, very good. And what bonds were those you were breaking?
Hydrogen bonds.
Okay, good. Show me where the helicase started when you were breaking it open.
On the top.
Okay, at the top. And the bonds, if you can, sort of put those bonds together. So, whenever a chromosome is ready to replicate, does replication start at the top or start at the bottom? What does it do?
I'm really not sure.
I think it starts at the top.
Okay, starting at the top, okay. Let's think, how many base pairs long is a chromosome? Is it just a few hundred, 1,000, 1,000,000?
A lot.
Okay, a lot. Okay, after y'all get through, now you only have how many?
8.
8. After you get through demonstrating your replication, think about, now you're only working with 8. If you had to go through 1,000,000, how much time would that probably take? So go ahead and keep working on yours and I'll come back. Okay, over here at PCR, I just got through talking to replication. Now they said they have a fancy enzyme called helicase. Do y'all have helicase? No. Okay, y'all don't have any helicase. Well, they said, oh, look at Leon. He would love for y'all to borrow his helicase. Leon, do they need helicase? Okay, why not? Because what?
They are not replication.
They're not replicating. Well, how are they going to open their DNA?
Heat.
The heat. Okay, very good, Leon. In PCR, how many different temperature changes do you go through?
3.
3. Okay, what's going to happen in the first one? Show me with your DNA.
It's going to get really hot and it's just going to open up.
Okay, good. So you don't even need an enzyme. How hot does it get?
94 degrees Celsius.
Okay, 94 degrees Celsius for this particular protocol. Do you know what that is Fahrenheit? Here, I'll see if this group can help. Let me ask y'all something for this group. Have y'all made your conversion 94 Celsius to Fahrenheit? What was it?
It was 201.2 degrees Fahrenheit.
Okay, so about 201 degrees Fahrenheit. Did y'all get about 201?
Yes, ma'am.
Okay, 201, and when does water boil Fahrenheit?
And 212.
212, so it's almost boiling. Okay, you got your DNA open. What are you going to do now?
Now it's going to cool down and primers are going to anneal.
Oh, primers? Nellie, what in the world's a primer? Is it just another segment of
Single-stranded DNA.
Okay. So, do y'all have a primer built, Matilda? Okay. Now, primer, that only is single-stranded. So is that RNA or is that DNA?
RNA?
RNA? RNA is single-stranded, but is that RNA?
Oh, no, because, yeah, okay.
What 4 bases do primers have on them? Do they have uracil? Now, it is temporarily single-stranded, so why is it single-stranded? What's the reason?
Because they're just going to match on the template strand.
Let's see.
On their amplifier.
You can hold it up. Maybe zoom just a little bit, not much. Okay, hold it up, and can y'all help him show what the primers do? What the primers do, Raymond? Okay, now and here's the other strand. Matilda, will you hold this one? Okay, so does the other primer, is it going to attach on the same side of DNA, or does it attach on the
One
other?
attach here and then one attach here.
Okay, good. So whenever we say we need primers, do we just need one primer, or is it actually a set? Oh, mercy, did we lose did one of our nitrogen bases take flight?
Yeah, that's good.
No, it's not one.
Okay, so that primer will attach up there. Okay, good. So primers attach, and then, so that's your second step, right? And what's the name for this step right here?
Anneal.
Anneal. What's your last step?
Extension.
Extension. Now, to extend, y'all haven't said anything about enzymes. Do y'all need an enzyme for that?
Yes.
And what's it going to be? Okay. Let me go see what enzyme Fruity has. What enzyme is that you have, Fruity?
Fruity has ligase
Okay, and what's its purpose?
to put together the Okazaki fragments.
Okay, and so these are the Okazaki fragments. Are they on the leading or lagging strand?
Lagging.
Lagging strand. Now this group back here has Taq polymerase that's going to do what for us, Matilda? What is it going to attach? Y'all can help her. Okay, the nitrogen bases. Who do y'all have to attach your nitrogen bases?
DNA polymerase.
DNA polymerase. Okay, y'all hold on. Y'all are using Taq polymerase to do the same thing they use DNA polymerase for. Why are y'all using Taq polymerase?
Because the DNA polymerase can't withstand the heat.
Okay, and how hot is it going to get?
72 degrees Celsius.
Okay, for the extension step, that's right. How hot does it get for replication?
37 degrees Celsius.
Okay, 37 degrees Celsius, which happens to be
Your body temperature.
body temperature. Okay, good. So, you said ligase attaches Okazaki fragments.
Yes.
And who puts on the nitrogen bases?
DNA polymerase.
Okay, good. Now, who puts on their nitrogen bases?
Taq polymerase.
Okay, good. So, how many different enzymes have y'all had to work with so far?
3.
3. Okay, good.
Initial denatured.
Okay, so y'all got your temperature conversion figured out?
Yes, ma'am, we did.
Okay, and then y'all are programming. If you open it up real quick for me, please. Oh, look, you happen to have, you can zoom just a little bit, not much, and then zoom back out. You happen to have a PCR tube in here. Do you see this little white bead? Okay, we use PCR ready-to-go beads. What are some of the ingredients y'all need in order to actually do what they're modeling over here? What's in this white bead that they would have at their table? Let me interrupt y'all just for a minute. Here's a PCR ready-to-go bead, and in this white bead, what's on your table that's actually in this white bead that you need if you're going to make an amplicon, a segment of DNA? Anything on your table? Y'all see anything on their table?
Yes, the DNTPs.
Okay, DNTPs, which are nucleotides, but why are they called DNTPs?
Because they still have the triphosphates.
They still have the triphosphates. Well, how come when we say a nucleotide, there's just one phosphate? What happened?
The other two are broken off
Okay, the other two were broken off for what reason?
to make energy to make it.
Who are you holding there, Ramon?
Taq polymerase.
Okay, does Taq polymerase need that energy? Who needs who's putting the nitrogen bases on? You? You're Miss Taq polymerase. So, in order to do that, it takes energy. So, where are you going to get the energy from? You're going to take it from those two phosphates where they have it stored in their bonds. Okay, so you have. What about what Ramona's holding? Taq polymerase. Is it in here? Is it in this little bead, Taq polymerase? Oh, the little beads pop to the top. You have to be careful sometimes. There it is. See, the little bead. Y'all saying, where's the bead? Okay, there's the bead. Is Taq polymerase in here? What do y'all think? Is it necessary for PCR? It sure is. So Taq polymerase is already in the bead. What else was already in the bead? That's right, your nitrogen bases, your DNTPs, and your polymerase. There's a couple of other things, but as far as what's on their table, that's there. It's hard when you're using these models to get everything that you need. But do y'all happen to remember, what do you call the friends of enzymes that help them do their job? Cofactors. So there's even cofactors in here. It was getting to be a lot, so you don't have cofactors at your table, but that's right. Okay, good. Y'all also had to do some math where you had to figure out how many nitrogen bases are going to be formed after a certain number of cycles. After 10 cycles. And first of all, name the 3 steps of a cycle, Mavis. Do you remember the 3? You said 3 temperature changes. You start out really high for denatured. You go down for anneal, and you come up for extension. Okay, and that's 1 cycle, right? Just after 1 cycle, how many copies of your target sequence do you have? 2. Okay. Now do that 10 times. Go through that 10 times. How many will you have?
1,024.
How'd you get that, Sue?
Because you did the 2 that you got, and you did it to the 10th power.
Okay. And so what was the how'd you get the 2? I understand taking it to the 10th power, but what about just the 2?
Because you have 2 single strands of DNA.
Okay, so from 1 DNA you get 2 copies after the first cycle. Is that right, Eunice?
That is correct.
Okay, good. Okay, so has everybody had practice programming the, okay, so Mavis is going. Oh, on Elvis. Okay, good. Okay. Well, while y'all are working on that, I'm going to let these two sort of compare what happens. So you can keep working on that, but I may ask you to follow something that we're going through. Okay, let me turn on the magic eye. Can you pretty much see everybody that's in the room? You can see the replication while they're working. Can y'all talk about what y'all were doing while I'm getting the magic eye ready?
Yes.
Yes, we can.
Right now we are separating our nucleotides, so whenever we reenact this whole thing, DNA polymerase will help me put these on there. That's what we're doing right now.